Hello everyone. Last lecture we had started with animal tissue, chapter animal tissue. In chapter animal tissue, we had studied in detail about muscular tissue, and in muscular tissue basically we had studied skeletal muscles. In today's lecture we are going to study two different more type of muscles, that is smooth muscles, or which is also called as non-striated muscle, and the other type of muscle we are going to study is cardiac muscle. So let us study smooth muscles or non-striated muscles. Now why it is called as non-striated muscle? Because they do not show presence of striations. Striations meaning bands. In skeletal muscles last time we had seen that it shows presence of dark band and light band. But here the smooth muscles, as they do not show any kind of bands, that's why it is called as smooth or non-striated muscles. Now these muscles, they are present in the form of sheets or layers. You can see they are present in the form of sheets or they are present in the form of layers. Each muscle cell is spindle shaped or fusiform. These are the muscle cells, okay, and these these smooth muscles or non-striated muscles, they are spindle shaped. Spindle shape means what? They are tapering at both the ends. Can you see they are tapering at both the ends? Whereas they are broad in the center. So such is called as spindle shape or fusiform. The fibers are unbranched, having single nucleus at the center. See in the diagram, they have a single nucleus in the center. Yes, and they are unbranched. Unbranched means what? They are single. They do not show any kind of branches. And hence these fibers, they are unbranched, having a single nucleus in the center. Sarcoplasm contains myofibrils. We had studied last time what is sarcoplasm. The cytoplasm of the muscle cells, they are called as sarcoplasm. So this is the cytoplasm. This is the nucleus. This is the cytoplasm. And cytoplasm is called as sarcoplasm. And sarcoplasm basically consists of myofibrils. See, these are myofibrils. So sarcoplasm basically consists of myofibrils. Myofibrils, they are made up of contractile proteins called as actin and myosin. Last time also we had studied that these myofibrils, they are made up of proteins. The two basic proteins are actin and myosin. Smooth muscle contains less myosin and more actin as compared to skeletal muscles. Smooth muscle contains less myosin. We had studied in last lecture that the dark bands, they are basically made up of more number of myosin, and hence they appear dark in number and thick also. But these smooth muscles, they have less myosin, whereas more amount of actin. Actin in skeletal muscles, it was made up of light band. The light band basically is made up of actin, right? So here in smooth muscles, you can see less myosin, whereas more actin as compared to skeletal muscles. Striations are present. Striations means what? Bands. The bands are present. That means no dark band and no light band. They undergo slow and sustained involuntary actions. They undergo slow. That means the contractions of the muscles are slow, and involuntary contractions. Involuntary means what? The movements which are not under our will, that is, which are not under our control. So that is why they are called as involuntary contractions. Now, where are basically these muscles found? These muscles are found in the internal organs, and the movement and the contraction of the muscles of internal organs, they are not in our control. And hence it is said that they undergo slow and involuntary contractions. They are innervated by autonomous nervous system. Innervated means what? They are supplied. Okay, they are supplied by autonomous nervous system. Now, what do we mean by autonomous nervous system? Autonomous nervous system, it is the part of nervous system that controls muscles of internal organs. When we see which are the internal organs, basically heart, yes, then we also have internal organs like lungs, stomach, intestine, blood vessels. Now, all the movements of these muscles of heart, muscles of stomach, muscles of intestine, they are not under our control. And hence, the movement of the smooth muscles, they are innervated by autonomous nervous system. That means the autonomous nervous system, which is a part of central nervous system, that is brain, that controls the movement. Where are they found? They are found in the walls of visceral organs. Visceral means what? Internal organs, and also in blood vessels. And hence they are also called as visceral muscles. Visceral muscles means what? Visceral means internal. And hence they are called as internal muscles. They may be arranged lengthwise, that is longitudinal muscles, or around circumference, that is circular muscles of any organ. In any organ, they can be found either in the lengthwise, that is in the form of longitudinal muscles, or in the form of circular or circumference, and hence they are called as circular muscles of any organ. Next type of muscle we are going to study is cardiac muscles. The name itself indicates cardiac. Cardiac means what? Basically the location, or where are they found? In the heart region. Now, muscles of this tissue shows characters of both striated and non-striated fibers. So when we see this muscle, these are the cardiac muscles. In the cardiac muscle, you can see certain bands, that is dark band and light band. So dark band and light band, it indicates striations, and hence these muscles of the tissue, they show characters of both striated muscles and non-striated fibers. Sarcolemma is not distinct. Sarcolemma means what? The membrane which covers the muscle fiber, it is called as sarcolemma, whereas the cytoplasm is called as sarcoplasm. So here the membrane, the protective membrane, the protective covering, which is called as sarcolemma, that is not distinct. That means not very much clear. Hence, uninucleate muscle fibers appear to be multinucleate. Hence, in the fibers where there is only one nucleus, that appears as, here you can see the sarcolemma is not distinct. Okay, so here that is why the fiber or the muscle fibers, basically instead of single nucleus, it appears having many nucleus. So that is called as multinucleate. Adjacent muscle fibers are joined together to give branched appearance to the tissue. This is one muscle fiber. This is another muscle fiber. So the adjacent muscles, they are joined together to give a branched appearance. Okay, you can see they are joined together by a branched appearance. Points of adhesion of muscle fibers are formed by a transverse thickening of sarcolemma, that is called as intercalated disc. As you can see, this is one muscle fiber, this is another cardiac muscle fiber. So they are joined together, okay, by a intercalated disc. What is intercalated disc? It is a transverse thickening of sarcolemma. Okay, so there are points of adhesion. Points of adhesion means what? Points of joining. This is one, this is another. So this is the point of joining, that is point of adhesion. And this point of adhesion, it is in the form of transverse thickening. And this transverse thickening, it is called as intercalated disc. Now, what is this function of this intercalated disc? These junctions at places allows cardiac muscles to contract as a unit. That means not a single cardiac muscle. Okay, these are the muscles. Okay, muscle fibers. These are the muscles. Okay, so all these cardiac muscle fibers, they contract together. Okay, all together, not one by one. So they contract together as a unit. So these functions, these junctions at places allow cardiac muscles to contract at the same time as a unit. The cardiac muscles are striated involuntary muscles. What are cardiac muscles? They are striated. They show dark band and light bands. Okay, if we see the myofibrils, they show dark band and the light band. So they are striated and they are involuntary muscles. Involuntary, that means the contraction is not under our control. That means we cannot control the heartbeat. Okay, so that is called as involuntary muscles. Some mammalian cardiac muscles are modified. That means they are capable of generating impulse of on their own, and hence mammalian heart is called as myogenic heart. Okay, now what does it mean by that? The cardiac muscles are capable of generating impulse on their own. If we study structure of heart in detail, we will come to know that in structure of heart there is a point of creating impulse which is called as SA node. So this SA node, basically they are the muscle which can contract on its own. It does not need external stimuli. So when we see some mammals like human being, okay, so they can generate impulse. The SA node can generate impulse. SA node is sino-auricular node, and this sino-auricular node, that is a type of muscle, it can generate electrical impulse on its own. It does not need external stimuli, and such type of heart which does not need impulse, such is called as myogenic heart. I hope you're getting it. That means they can generate impulse on its own. They can contract on its own. That is such type of heart. Mammalian heart, it is mostly found in mammals, and hence such mammalian heart is called as myogenic heart. Whereas in some animals, cardiac muscles needs neural stimulus to initiate the contraction. Neural stimulus means what? They need a stimulus from the central nervous system. Okay, they need the stimulus from the central nervous system. What happens? The central nervous system, it produces certain type of hormones, okay, from the endocrine gland. But the endocrine gland, it needs stimulus, and that stimulus is given by the central nervous system. Unless and until the endocrine gland does not get stimulation from the central nervous system, neural hormone will not be produced. Okay, so such is called as neural stimulus. So until and unless neural stimulus is not initiated, the contraction will not occur. Such a heart is called as neurogenic heart. So that's it for today's lecture. Okay, thank you very much.